Okay, good morning, friends. Rather good afternoon. So, in the yesterday's class, we discussed about respiration. So, what is respiration and how actually the glucose gets decarboxylated and dehydrogenated and ultimately giving rise to water and carbon dioxide and energy, wherein we have seen the breakdown of glucose occurs at three phases. First phase is called what we call glycolysis, wherein glucose molecule splits into two molecules of pyruvic acid. And this pyruvic acid, in the presence of oxygen or in the absence of oxygen, it takes it two routes. In the presence of oxygen, of course, it makes an entry into mitochondrion. In the mitochondrion matrix, the decarboxylation and dehydrogenation of pyruvic acid takes place. But before pyruvic acid, pyruvic acid undergoes decarboxylation, dehydrogenation in the presence of a multi-factor enzyme and ultimately giving rise to what we call acetyl coenzyme A, 2-carbon compound. And this is the connecting link between, glycolysis and Krebs cycle. This 2-carbon acetyl coenzyme A combines with 4-carbon oxaloacetic acid. Of course, we get a 6-carbon, what we call citric acid. As the citric acid is 6-carbon, hence it is called a citric acid cycle, or as it has 3 carboxyl groups, it is also called a TCA cycle, tricarboxylic acid cycle. And this, 2-carbon acetyl coenzyme at two places, it loses its carbon and it generates what we call carbon dioxide. And in all, at 4 points, the dehydrogenation takes place. At 2 places, decarboxylation takes place. Thus, in all, 5 molecules of NADH2s and APADH2s and 2 molecules of GTP are produced. And we have seen the energetics, what we call the energetics of this glycolysis and Krebs cycle, wherein we have seen in glycolysis, the net gain is of only 6 ATPs, and whereas in glycolysis, whereas in Krebs cycle, we get 30 ATPs. So in all, we get 36 ATPs when one glucose molecule is digested or completely oxidized. And of course, this energy will be used by cell. And as the cell organelle, that is, mitochondria is involved in the production of ATP. Hence, mitochondrions are called powerhouses of the cell. This we have seen. And of course, if there is no oxygen, the pyruvic acid, undergoes partial oxidation. And based on the end product, this we call the partial oxidation of pyruvic acid is also otherwise called fermentation. Now, based on the end products of fermented end products, it could be alcoholic fermentation or oxalic acid fermentation or citric acid fermentation or lactic acid fermentation. See, for example, in curdling of milk, we get lactic acid. This is lactic acid fermentation. So this fermentation end products are carbon dioxide and ethanol if it is alcoholic fermentation. And these two byproducts of fermentation are very useful. I told you yesterday, the ethyl alcohol is used in distilleries, making of these beers and all, alcoholic beverages. The ethyl alcohol is used. And whereas the carbon dioxide is used by bakeries in the production of, bread. This we have learnt in in yesterday's respiration. And today we will learn about the nitrogen metabolism. You know, in our earlier classes, while dealing with mineral nutrition, I told you there are a number of elements that are essential for the healthy growth, reproduction of the plant, and based on the quantity of the particular element needed by the plant, it is of what we call a macronutrient or micronutrients. And of course, we have seen there are 9 macronutrient, and there are 7 or 8 micronutrients. Of these 9 macronutrients, nitrogen is the most important. So carbon, hydrogen, oxygen, of course, these are 3. And the next comes the important 3, nitrogen, NPK, what we call nitrogen, phosphorus, and potash. These 3 are very, very important. And of course, and to get maximum yield, farmers replenish, because as intense cultivation is going on, when intense cultivation is going on, there is a depletion in the nitrogen from that particular field. So the loss of nitrogen from the field occurs 3 ways. One is by absorption by the crop plants. Second one is what we call leaching. Entry of the nitrogenous products and nitrates, ammonia, deeper layers below rhizosphere, that is below your root system. And the third one is loss of nitrogen occurs by denitrification. This is another process. Okay, so that is how even the precious nitrogen is lost in the form of elemental nitrogen. This we call denitrification. Okay, so now if you closely see the nitrogen, you will see there is a kind of cyclic movement. There is a cyclic movement of nitrogen. So nitrogen, the ultimate source of nitrogen is atmosphere. The atmospheric nitrogen content is, as you know, it is 78%. Though atmosphere has 78% of nitrogen, but of course, this is not available. As if somebody sailing on high seas, everywhere you will see water. Water is present everywhere. If you are traveling on a sea, water is present everywhere, but that water is unfit for consumption. We cannot take that because of its high salt content. So also here, our atmosphere has a lot of nitrogen. As you know, 78% of the atmospheric gases, nitrogen accounts for 78%. So though large quantity of nitrogen is present in and around the organisms, especially plants, but plants are incapable of using this elemental nitrogen. The importance of nitrogen. Nitrogen is a very essential element. Of course, we discussed in detail. See, nitrogen is a constituent of so many important biomolecules. Say, for example, chlorophyll. Nitrogen is there. And of course, nucleic acids, purines, pyrimids, enzymes, proteins. So whatnot, all biomolecules. So enzymes, proteins, nucleic acids, chlorophyll. In all of them, nitrogen is an important constituent. So hence, the importance of nitrogen cannot be underestimated. So now, as I told you, if you closely observe, though 78% of nitrogen is present in the atmosphere, but it is not in useful form, or our higher plants or our crop plants are unable to take up this nitrogen that is present in large quantity in the atmosphere. Okay, so but there are certain microorganisms. Certain microorganisms are capable of converting this elemental nitrogen into useful form, that is maybe to nitrate form or to ammonical form. So these are the 2 forms in which nitrate form and ammonical form plants absorb their nitrogen requirements ultimately from the soil. So the nitrogen that is present in the atmosphere has to reach the soil. From soil only plants absorb. But of course, after absorbing plants, what they do? They grow, they reproduce, they give us seeds and fruits, etc. Human beings, all animals depend on plants for their nutrition. So, of course, all plants and animals, after leading their life, has to die. Each and every organism, for that matter, dies. All the organisms that take birth have to die at one time or the other. So when the plants and animals that live on nitrogen, if they die, so whatever the nitrogen that is there in their body has to be released. It has to be released. If there is no release, generally you will see if organisms like bacteria, fungal are not there. So even by this time, the entire surface of the earth would have been occupied by the dead bodies of animals and plants and human beings. But that is the case. There are so many microorganisms that are present in the soil, in the atmosphere, that break these complex molecules and convert them into simpler molecules by degradation, by putrefaction. Okay, so this process of breaking the complex molecules into simpler molecules and thus releasing nasty ammonia. If an organism, if any cat, rat dies in the vicinity of our house, we feel after 2 or 3 days, we see pungent smell coming, maybe H2S or ammonia, etc. So that is coming because of the action by various microorganisms, various bacteria that are there in that area. So these bacteria, these fungal groups, they break these complex molecules and ultimately release the gases into, of course, atmosphere and the minerals into the soil. This is what we call mineralization. This is what we call mineralization. So when organisms die, the complex molecules present in them are broken by microorganisms and ultimately resulting in the release of ammonia. Hence, this process we call ammonification. So the ammonification is a process wherein the dead and decaying organisms are the complex molecules are broken into simpler molecules and ultimately ammonia is released. This we call ammonification. So this ammonification is done by soil microorganisms and the organisms that are present in aerial organisms. So now this ammonia that is released, some amount of ammonia enter into the atmosphere, the other ammonia that is released by the microorganisms will combine with various, minerals, especially calcium, magnesium, sodium, etc. And of course, their salts are formed. So calcium, ammonium nitrate, sodium nitrate, etc. So they are formed. So that is how the ammonia becomes salts, nitrogen salts, calcium nitrate, potassium nitrate, sodium nitrate, etc. are formed inside the soil. So now this nitrate form, of course, will be converted by chemoautotrophic bacteria, what we call nitrifying bacteria. So the ammonia that is present in the form of salts will be oxidized. Ammonia by two types of organisms. for example, nitrosomonas and nitrobacter. Nitrosomonas converts ammonia into nitrate form, and this nitrate is further oxidized to nitrate by nitrobacter. So this is how the nitrogen will be released into the or into soil, and this nitrate form will be easily absorbed by the plants that are living, especially the roots. Through the roots, again, plants will be able to absorb this nitrate form. So this, of course, nitrate ammonia will be converted into nitrate, but there are some microbes that that further degrade nitrates into dinitrogen form. See, the microorganisms, especially Thiobacillus denitrificans, is a bacterium that converts the useful fertile nitrate form, that is nitrate form, into dinitrogen N2 form. This we call denitrification, conversion of nitrate into elemental nitrogen form or gaseous nitrogen form. N2 form is called this denitrification by Thiobacillus denitrificans. Okay, so that is how nitrogen is again released into the atmosphere. We have seen from atmosphere the nitrogen. Now the atmosphere, the nitrogen that is present in the atmosphere will be absorbed by, especially, or will be fixed. Of course, I told you there are certain microorganisms which are capable of fixing this nitrogen. This way, this organism is called nitrogen fixing organisms. Nitrogen fixing organisms are primarily, cyanobacteria and the bacteria. The bacteria are, again, I'll tell you in detail later. So the bacteria, especially the aerobic bacteria, anaerobic bacteria, free-living bacteria, and what we call symbiotic bacteria. So these are some of the bacteria that are capable of converting atmospheric nitrogen into ammonia form. This conversion of nitrogen into ammonia form is known as nitrogen fixation. Nitrogen fixation. So if you carefully observe, so starting from atmosphere, nitrogen will be absorbed by nitrogen fixers, this biological nitrogen. But in nature, there are two other ways of nitrogen fixation. One is what we call physical nitrogen fixation, that is during thunders, during lightning, during high temperature and pressures. When lightning occurs, the elemental nitrogen, that N2 form, breaks, N triple bond N breaks, and you get nitrogen atom. This nitrogen atom may combine very unstable. The very unstable nitrogen may combine with hydrogen and may form ammonia, or this nitrogen may combine with oxygen and form nitric oxide. The nitric oxide may combine with another atom of nitrogen, and ultimately nitrates are formed. So this is fixation by natural forces, that is during lightning, during thunders, the nitrogen breaks and atomic nitrogen formation takes place, and this may combine with oxygen, or in some cases this elemental atomic nitrogen may combine with, in stratosphere area, may combine with ozone, and of course you get nitrates, and this may combine nitrogen oxides, and these nitrogen oxides may combine with water vapor, and ultimately nitric acid may be formed. And when there are rains, the nitric acid along with the rain reaches soil and combines with various calcium, magnesium, minerals, and ultimately their respective mineral salts are formed. So what I mean to say is, if you observe very closely, the nitrogen shows a kind of revolving. This revolving of nitrogen from atmosphere to organisms, from organisms to again back is what we call nitrogen cycle. So in terms of what we call, in which nitrogen is shuttled back and forth by microflora is what we call nitrogen cycle. So this nitrogen cycle is, you may, not very difficult, but it is an important question. Maybe asked under short note or maybe under, essay. If it is a nitrogen cycle, it becomes an essay, or short notes, ammonification, nitrification, denitrification, nitrogen fixation, like this, they may ask. Okay. Now with this background, now we'll move to various important processes regarding nitrogen conversions that take place in the organisms. Say, so you see, I told you nitrogen is very, very important, and this nitrogen will be absorbed by the plants only mostly in two forms. One is ammonium ion form and also nitrate form. So this this form, this ammonia form. So this nitrate, if it is nitrate form also, it has to be converted into ammonia. So only in the ammonium ion form, this ammonia will be taken up by various dicarboxylic acids and ultimately amino acids are formed. So this assimilation of nitrates, this assimilation of nitrates into cytoplasm is what we call metabolic reduction of nitrate. So here the nitrates that are present, they are to be reduced. Nitrate is in oxidized form. I told you nitrate in oxidized form will be converted into ammonia at two steps. At first step, nitrate will be converted into nitrite, and in the second step, nitrite will be converted into ammonia. So there are two important enzymes that are involved in the conversion of nitrate into ammonia. One is nitrate reductase, the other is nitrite reductase. Say, for example, nitrate reductase. This nitrate reductase, so here simply you can see nitrate NO3. NO3 plus 2 electrons and 2 protons, you get what we call nitrite NO2 plus H2O. See, NO3 is there getting converted to NO2. So the oxygen atom that is there combines with the 2 protons and ultimately you see liberation of one molecule. So here for this to occur, ATP is very much needed. So, and this in the second step, generally the ATP and the protons and electrons for the nitrate reduction will be supplied by, ferredoxin. We read about ferredoxin, in cyclic photophosphorylation relation from second photosystem to photosystem 1. Then we have between two, we have quinone, and photosystem 1 again, the electrons will be moving to ferredoxin, and this ferredoxin, converts NADP into NADPH2. So here for the reduction of nitrate into ammonia, this is a two-step I told you. In the first step, nitrate will be converted into nitrite in the presence of nitrate reductase. For this, 2 electrons and 2 protons are needed, and ultimately there is a release of one water molecule. And in the second step, the nitrate is NO3. So the nitrite will be further reduced to ammonia form by here the ferredoxin. The reduced form of ferredoxin, in the presence of reduced form of ferredoxin, NO2 plus ferredoxin plus 8 protons ultimately change to what we call ammonia, NH4, ferredoxin oxidized, and of course 2H2O. See, I am giving you 8 protons. NO2 I say. NO2 means from N we are getting NH4. That is 4 protons are used. Another 4 protons will be used for converting. This O2 is there, this O2 ultimately gets converted into 2H2O. So thereby, so here NO3 minus 2 electrons, 2 protons, you get nitrite in the presence of nitrate reductase, and in the further oxidation, the nitrite reductase converts the nitrite into ammonia form. So this what we call nitrate reduction. This process we call nitrate reduction. So simply what is happening? Nitrate is NO3 form is getting converted into ammonical form. This we call nitrate reductase, and generally the nitrosomonas and nitrobacter are the bacteria, and they have what we call two important enzymes, nitrate reductase and nitrate reductase, that will be used for reducing nitrate to. So second is, now of course ammonia is available, ammonical form is available. Now this ammonia has to be integrated into ammonia, these amino acids. The ammonia has to be integrated. The integration of ammonia into or conversion of ammonia into amino acid occurs. So amino acids. So in each amino acid, of course, we have one amino group and one carboxylic group. One amino group and one carboxylic group is there. This we call amino acid. So amino acids, there are 20 amino acids are there, and amino acids are the building blocks. They are the building blocks for polypeptides, and these polypeptides in turn change to proteins. And of course, the polypeptides, of course, give rise to what we call enzymes. So all enzymes are proteins, of course. So the protein, in proteins and in enzymes, the amino acids are the building blocks. So for the synthesis of amino acids, what we need? Ammonia form. So this integration of ammonia takes place by in the presence of different enzyme systems are there. See, for example, one keto acid. So these keto acids, if you remember the glycolysis. In the glycolysis, we have seen so many intermediary acids. Intermediary acids we have seen. For example, we have seen oxaloacetic acid, we have seen alpha-ketoglutarate acid, we have seen succinic acid, we have seen malic acid, we have seen. These are all the dicarboxylic acids or tricarboxylic acids. Carboxylic acid, simply. They are the carboxylic acids. So when the carboxylic acid takes in ammonia, it gets converted into a amino acid. For example, a 5-carbon alpha-ketoglutarate acid, if it gets ammonia, what we call reductive aminase, this we call. In the presence of reductive aminase, the ammonia gets incorporated into a carboxylic acid, and a respective amino acid is formed. If it is, for example, if it is alpha-ketoglutarate acid, you get a 5-carbon amino acid by name glutamic acid or glutamate. See, for example, if it is a 4-carbon, it is a 4-carbon oxaloacetic acid, and you get what we call aspartic acid. So malic acid, malate. So like this, aspartic acid, and further amination, it gives rise to what we call an amide called aspargine. If glutamic acid or glutamate takes one more amino group, it leads to formation of what we call amides. In amides, so in amino acids, you have one amino group. If it is an amide, you have 2 amino groups. Amides. Generally, amino acids, they are toxic, and they cannot be stored. So generally, the amino acids are converted into amides. Amides are not lethal. They can be stored for longer duration. So now how this amino acid synthesis takes place? We will learn. So generally, the amino acid synthesis occurs by two processes. One what we call reductioamination, the other is called transamination. Transamination is very simple. Transamination means, see, if, for example, we have seen in glycolysis, glucose plus ATP, we get what we call glucose 6-phosphate. And glucose 6-phosphate, for example, fructose 6-phosphate. Phosphoenol pyruvic acid. So there is what is happening. It is either ATP is added or phosphorylated or dephosphorylated. So here also aminase or deaminase. So reductioaminase is wherein amino group is added. That is called reductioaminase. Transaminase means, for example, a keto acid and amino acid is there. The amino acid gives its amino group to keto acid and thereby converting the keto acid into amino acid. By losing its amino group, it becomes a keto acid. So when amino acid and the keto acid are, are combined in the presence of ATP, the amino acid changes to keto acid and keto acid changes to amino acid. This we call transamination. So there is a transfer of amino group. There is a transfer of amino group. This we call transamination. Then the other most important is reductioamination. Reductioamination is a process wherein the amino group is added to a keto acid, converting it to a. So here, not one keto acid, one amino acid. Here we are adding amino group to the keto acid. Thereby, keto acid becomes an amino acid. This what we call reductioamination. This reductioamination, for the reductioamination to take place, two important enzyme systems are there. And this what we call, in the first step, what we call, this synthesis has been worked out. So initially people thought that the keto acid directly incorporates amino acid and turns itself into a amino acid. But later on, the detailed investigations revealed that no, it is not the keto acid that takes amino group. So by using radioactive amino groups, scientists, especially the Mifflin and Lee, they proved beyond doubt that it is not the keto acid that is the acceptor amino group, but it is an amino acid that takes the amino group, thereby converts itself into an amide. And this amide combines with a keto acid and thereby losing its amino group, you get two amino acids. So here amide has two amino groups. When you combine amide with a keto acid, by losing its one amino group, the keto acid becomes amino group, the amide becomes an amide. So like that, at the end of reductioamination, you will see formation of two amino acids. Of course, this occurs, this in case of transamination, of course, you see, for example, if you start with a glutamic acid and if a glutamic acid and a glutaric acid and an aspartic acid and oxaloacetic acid are there, the oxaloacetic acid changes to aspartic acid, where glutamic acid changes into a glutaric acid. That what I mean to say, glutamic acid has amino. This glutamic acid gives its amino group to oxaloacetic acid and thereby converts oxalic acid into aspartic acid, and glutamic acid, by losing its amino group, becomes a alpha ketoglutaric acid. So this is what we call the reductioamination and transamination processes. Then nitrogen fixation. I told you in nitrogen cycle, the nitrogen fixation. I told you aminification, nitrification, denitrification, and the important other process is what we call nitrogen fixation. This is the most important thing of all. This is the most, of course, every step is important, because if one is missing means the cycle gets broken. it will have, disastrous effects on organisms. So that cycle has to be continued. Each and every link is very important. If a chain is there, for example, a grand chain is there. If one link is lost, means it loses its identity. It cannot stand. So each and every link is very, very important. So also, even in nitrogen cycle, each and every step, ammonification, nitrification, denitrification, and nitrogen fixation, all are very, very important. So now we will learn about this nitrogen fixation. I told you already, we discussed it. There is 78% of nitrogen is there, but our crop plants are unable to take this atmospheric nitrogen and use them. Only there are same microorganisms, small microorganisms, which have the ability to convert this, nitrogen, elemental nitrogen or gaseous nitrogen into a fixed form, that fixation form. So this fixation, especially into ammonical form. So now this fixation, I already told you, this nitrogen fixation is done by three processes. One is what we call, a natural process or physical process, and the other is biological process. There is one more process, what we call industrial. See, all our nitrogen fertilizers, what we get, your Gromor, your DPT, all these, all our chemical fertilizers. They are produced in factories. So now this chemical nitrogen production, nitrogen fertilizer production involves, under very high temperature and pressure. When you subject nitrogen to very high pressures, as high as 1,000, atmospheric pressures, and more than what we call of 400 to 600 degrees of temperature under such high pressures and temperature, the elemental nitrogen, the nitrogen triple bond nitrogen breaks, and you get nitrogen atoms. And this nitrogen atom under high pressure and high temperature combines with hydrogen, and thereby you get ammonia. This is industrial nitrogen production. This industrial nitrogen production is an important process, and that is being done in all our factories. Thereby, we are able to meet this production, what we call Haber-Bosch method. So Haber-Bosch method is a method wherein under 1,000 bars of pressure and temperatures of 450 degrees centigrade, the nitrogen fixation takes place. Under high temperatures, the nitrogen and hydrogen combine, and you get ammonia. This physical nitrogen, I already told you, under high temperature, under lightening conditions, the nitrogen combine with oxygen, nitric oxide, nitrogen dioxide, nitrate, and of course, combination with nitrogen oxides with water vapor, you get nitric acid, and nitric acid comes down along with the rain and combines with whatever the inorganic metals that are present in the soil, and ultimately their salts are produced. Now, the biological nitrogen. So we know the chemical fixation, that is industrial. Then, of course, physical. The third one, most important, is biological nitrogen fixation. This biological nitrogen fixation is very, very important. And there are only, I told you, only few microorganisms that are capable of converting this atmospheric nitrogen into ammonia. They have an important, enzyme. They, all the nitrogen fixers, they have an important enzyme in their cells called nitrogenase. There are separate genes responsible for the synthesis of this nitrogenase, and those genes involved in nitrogenase production are called NIF genes. NIF means nitrogen fixing genes. So these NIF genes are present only in certain legumes, and especially in case of what we call the rhizobium and other bacteria, they have this mechanism in their cells. They have this nitrogenase enzyme, and this will be produced by genes that are code for this nitrogenase enzyme. So this important nitrogen fixers, for our convenience, we can divide them into what we call asymbiotic bacteria and symbiotic bacteria. So under asymbiotic, they are again aerobic and anaerobic. Aerobic, for example, Azotobacter is there, and Azospirillum is there, and Klebsiella. These are all asymbiotic and aerobic. Then anaerobic. Anaerobic are, see, asymbiotic means they are free living. Symbiotic means they make an association with their host. Good example for this is root nodules of legumes. Root nodules of legumes, there is an association with two organisms living together for mutual benefit is called symbiosis. The good example is root nodules of legumes. But whatever we are speaking now, learning is asymbiotic. Asymbiotic means they don't have any association with the host. They are free living. And these free livers are maybe aerobes or anaerobes. Aerobes are Azotobacter, Azospirillum, Klebsiella is there. Anaerobes, for example, Clostridium is there, Chromatium is there, and Thiobacillus are these some of the free living anaerobes. And of course, the other most important group of these bacteria that fix nitrogen are cyanobacteria, otherwise called blue-green algae. The blue-green algae, all the three forms, that is cellular, unicellular forms, filamentous forms, and also filamentous, heterocystous, and non-heterocystous forms are capable of fixing this nitrogen. As the research is going on, scientists are able to, find out what are the organisms that possess this nitrogen fixing genes in them and are capable of converting this elemental nitrogen into, this nitrate or ammonical form. So cyanobacteria, for example, unicellular bacteria are Aphanothes and Gliocaps are there. If it is a filamentous heterocyst, this is very, very important. Filamentous heterocystous forms are most efficient nitrogen fixers than non-heterocystous. Even non-heterocystous are capable of fixing, but the efficiency of heterocystous forms is more when compared to non-heterocystous. See, heterocystous forms are, you learned about them, Nasta cannabina are there. For example, filamentous non-heterocystous are Acinetoria is there, Plectonema. These are some of the cyanobacterial blue-green algae that are capable. In addition to this, we also have what we call Allosirrhizae, Tolipothrixis, Allosira, Tolipothrix, Cylindrospermum. These are some of the blue-green algal members that are capable of fixing, nitrogen. Many are there. Then we move to symbiotic. Symbiotic nitrogen fixers, whenever I say, I told you already, the legumes. The legumes are capable of, fixing nitrogen. And this association we called symbiotic. And generally, legumes are there. Each legume has its own specific nitrogen fixing bacterium. See, for example, Phaseolus vulgaris, our green gram, wherein the nitrogen fixing species is Rhizobium phaseoli. For example, Pisum sativum, what we call Rhizobium leguminosarum, is the bacterium, rhizobial bacterial species that is capable of efficiently fixing nitrogen. Soya bean, for example, Glycine max, Bradyrhizobium japonicum is the rhizobial species that is capable of fixing nitrogen in legumes like Pisum, Phaseolus, and Glycine. Then, of course, non-legumes. There are certain non-legumes where the microorganisms are capable of fixing nitrogen. Non-legumes, for example, your Casuarina. In Casuarina, Frankia is a bacterium that is capable of fixing nitrogen, Actinomycetes. And of course, ferns. In Azolla, a water fern, wherein Anabina azolla is capable of fixing nitrogen. And of course, Cycas, you learned in cycles we find specialized roots called coralloid roots, wherein you find Anabina and Nasdaq. Of course, certain fungi are there, especially fungi and algal association gives us what we call lichens. So certain in lichens, they have certain fungi and certain algal members like Nasdaq, which associate and give rise to a lichen, and that is capable of fixing nitrogen. So of all organisms, the rhizobium in case of legumes and cyanobacteria, free living bacteria, these two are two important groups of organisms that are capable of fixing nitrogen on a large scale. They're more efficient. Say in cyanobacteria, I already told you. So the families of Nostochaceae, Cytomanaceae, and Rivulariaceae, Strigonemataceae are capable of fixing Acidoria, Formate, Glucoccus, Anabina, Nasdaq. These are some of the important Tolipothrix, Alocera, Cylindrospermum. These are cyanobacteria that have the efficient mechanism of nitrogen fixation. So now this how do the mechanism of fixation? How this this mechanism for the nitrogen fixation, the mechanism is there is what we call enzymes, dinitrogenase. Dinitrogenase reductase is the enzyme. Nitrogenase, of course, it has two parts. Nitrogenase is an enzyme, and that has two parts. One part is bigger part, has what we call molybdenum, iron, and sulfur, and the smaller part has called iron, sulfur, and A2B2. These are the dinitrogenase, and these various cofactors and cofactors having molybdenum, iron, and sulfur, they regulate the movement of what we call protons and electrons, and ultimately convert this nitrogen into ammonia form. So here generally in all organisms, so the maybe you may be your roots or maybe in leaves, etc. Here the peridoxin is the electron supplier. So from peridoxin, the ferredoxin, by using ATP in the presence of magnesium, supply electrons to what we call dinitrogenase oxidase. And this nitrogenase, dinitrogenase oxidase, by taking electrons from ferredoxin, gets converted into in reduced form, dinitrogenase reduced form, and this dinitrogenase enzyme, again the electrons move to what we call elemental nitrogen, and this elemental nitrogen, by taking protons and electrons from ferredoxin and dinitrogenase enzyme, ultimately converts elemental nitrogen into ammonical form. So here, for example, if you start with nitrogen, N2 gets converted into ammonia in the presence of 8 electrons and 8 protons and 16 ATP and magnesium atoms. In the presence of magnesium atoms, you get what we call 2 ammonias, a molecule of hydrogen is produced. So here, in addition to ammonia, even hydrogen is also one of the end products in nitrogen fixation. So now this does and one more important thing you have to remember, the nitrogen is very, very sensitive to oxygen. If it is exposed to oxygen, it gets denaturated. It will not function. Hence, inside what we call heterosis, there is a compartmentalization wherein only limited amount of oxygen is available, and that oxygen will be used for oxidative phosphophosphorylation, whereby you get ATPs, and this concentration of oxygen is in such a low quantity that it will not harm nitrogenase, that is very, very essential for converting elementary nitrogen into ammonical form. So this is how the nitrogenase works or ultimately converts the elemental nitrogen into ammonical form. So this is about briefly, because this is a very big topic, generally in regular classes for this nitrogen metabolism, we need more than 5 or 6 classes. But within 1 hour, had to rush through, and I think uh, you learned something. If you have any doubt on this today's topic or the topics already covered, you can raise some of the your doubts. I'll try to clear them. Say, 2 more classes. Of course, tomorrow, same time, we have a one more class from 3 to 4 p.m. And in tomorrow's class, we will learn about what we call the plant growth, and especially growth regulators, otherwise called phytohormones, and the physiology of flowering, where we will discuss about the growth curve, what we call S-shaped curve, log phase, log phase, lag phase, and of course stationary phase S-shaped curve we'll learn, and also the important growth hormones. Of course, hormones are divided into growth promoters and growth retarders. Of course, we discuss about them such as growth promoters such as auxins, gibberellins, and cytokinins, and also the what we call the fruiting hormone. Of course, what we call the hormone that nowadays, now this is the season for mangoes. For mango ripening, we need certain hormones, and the important hormone that helps in ripening of fruits is called ethylene, a gaseous hormone. So that way we learn about oxygen, gibberellin, cytokinins, and phyto, and of course the ethylene, and also growth retardants also. We will learn about them in tomorrow's class. So by tomorrow, we will be completing the physiology part, and of course, whatever you know, maybe 2 or 3 classes we may get. Now we will try to. Of course, we lost 2 classes last. Saturday and Sunday we lost 2 classes. I think we don't know whether they will give one more chance regarding compensation for the classes that are lost, or else we may get only one class, which is on 2nd, I think. So in that class, we will discuss about the ecology part, wherein we discuss about ecosystem, the producers, the consumers, and of course, Xerox succession, hydrox succession, pollution, this and that. We will try to learn about concepts of ecology and ecology topic in the ensuing classes. But tomorrow we'll discuss about the concept of growth and concept of growth hormones. I think you're not raising any question now, so now it is time for us to close. So thank you for your patient hearing. We will meet tomorrow same time. Until then, bye.